Hi everyone, this is our final video for the semester, and it will be about metabolic channeling and metabolic pathways in the fasting liver. So just kind of tying everything together at the very end and putting, really emphasizing the linkage between all of the pathways we've talked about. So far throughout the semester, we've talked about quite a few pathways. Let's separate page for this. We've talked about, carbohydrate metabolism. We've mentioned, glycolysis going into the Krebs cycle. Krebs cycle is kind of like the centerpiece of everything, right? So we got like this little circle here of the Krebs cycle. And there's glycolysis going into it. But we also could go up into glycogenesis instead with our carbohydrates rather than running glycolysis and the Krebs cycle. We've talked about lipid metabolism. So we have synthesis of our fatty acids that we've talked about, and that's an offshoot of the Krebs cycle. We also have kind of the other way where we have beta oxidation, where we're breaking down our lipids to get acetyl-CoA to go into the Krebs cycle. And that's dependent on a pathway that came off of glycolysis, which was the pentose phosphate pathway that gave us NADPH, which we needed for our lipid synthesis, right? And don't worry about copying this little diagram down. This is just for illustrative purposes and not very good illustrative purposes. Apparently, this diagram is not so neat. I just want to show you just all together that we've done carbs, we've done lipids. We can break them down. We can, build the carbs up. We can break down the lipids. We can create the lipids. And then also protein metabolism or amino acid metabolism, where we're, having some proteins enter into the Krebs cycle at various stages, or we can have transamination or just plain old amination and create proteins, so protein synthesis. With our proteins, we have our glucogenic proteins when we're breaking them down, or we have ketogenic proteins. So the glucogenic could be used for gluconeogenesis. We can also have gluconeogenesis coming right off the Krebs cycle, or from glycolysis from pyruvate or from oxaloacetate. The ketone bodies, those are used for a million different things. They can be going into acetyl-CoA, or they can be, directly oxidized. So we have all of these things being just completely linked together. And for a lot of them, the Krebs cycle is sort of the hub, right? Like the main station here where we branch off and we can enter in or we can go off. Remember we talked before about, different shunts that we have. This is where we're going, just this relationship, everything all interconnected and intertwined within the cell. That's what metabolic channeling is. And this is not a new word for everyone. This is a word that we've heard before, right? And it just has to do with what is the main reaction, or what reactions are happening inside a cell. And that's driven by what enzymes we have, what substrates we have, how much of each of those are they doing negative feedback? So inhibiting other processes, stimulating other processes. So basically enzymes. So are we inhibiting or stimulating enzymes? All of those things are going to be determining which way we go. You know, are we going to be doing more Krebs cycle? Or are we going to be doing more glycogenesis or gluconeogenesis? Or, you know, what are we doing? And really, essentially, the main questions or the main situations a cell can have would be having lots of energy. Well, then you want to go into energy storage sorts of pathways. Lipid synthesis. Protein creation. So growth sorts of things. Anabolism. Creating cellular structures, saving energy, all that kind of stuff. Or we could be in the opposite situation where we're having low energy, so like the starvation state. Then we want to be doing energy creation pathways. We want to be breaking down stored energy. So we'll be doing lipolysis, proteolysis, catabolism generally. So breaking things down for energy. And each of these states is going to have different products, different substrates, different enzymes that are a result of that state that will drive us in different directions. So this picture here is just showing, you know, essentially what I just drew, but less, you know, messy in a way. So we just have glycolysis, showing that you could go into glycogenesis over here. You can, you know, break down the glycogen and create glucose and, you know, go that way, like that way. So it's all different ways, and each different way is going to be dependent on these factors. You know, what does a cell need? What does it have? How are those things interacting? And the idea of metabolic channeling also has to do with the idea that enzymes essentially carry one intermediary product to the next step or to the next enzyme or the next location of where they need to be in order to progress the pathway or to follow through on the pathway. So each enzyme in the step carries the intermediary products or substrates to the next step. So it's all very organized, all very logical. You know, like what we talked about in our previous video, if you have fumarate present from the ornithine cycle, but you need to run the Krebs cycle, but you've run out of oxaloacetate. You need to take that fumarate and you need to convert it into malate, and then you need to convert the malate into oxaloacetate. And there will be an enzyme for every step of the way. You have to have each of those enzymes, but each one is going to create the necessary intermediary to go into the next step. So they're all dependent on each other, working together. Now this one, hard to see, very small. This is essentially showing a fatty acid channeling, and it's specifically looking at palmitate, which little tiny word down here. So for the words you need to really see, I'll write them out again. Otherwise, just follow along with the little chain. I know it's too small to see on this video, but palmitate is a fatty acid, right? So we know from our fatty acid synthesis from Unit 2 that we're going to be taking acetyl-CoA, we need NADPH, we need, you know, malate and malic enzyme and all that kind of stuff. We're not getting into all of that detail. We also need fatty acid synthase, we need de-elongation enzymes. All of those things are steps in that particular pathway. But where this comes into metabolic channeling, or how we can link this to metabolic channeling, is that here we have acetyl-CoA carboxylase. Let me write that down so you can see that. Acetyl-CoA carboxylase is an important enzyme in this process. It's the one of the initial enzymes in the process. But this is going to be essential for even starting the process by creating malonyl-CoA out of acetyl-CoA. And from there, we enter into using our fatty acid synthase and our fatty acid elongases, which we have several, right? So we can just elongate it kind of one time, end up there, or we can keep going and going and going and going and going and create all sorts of different kinds of fatty acids. But here we're just going to stop. We're going to look at just palmitate. Palmitate, all by itself, is an inhibitor of acetyl-CoA carboxylase. So palmitate inhibits essentially itself. So what this is doing is a sign. It's an intracellular signaling sign, really. You're creating palmitate, you've made fatty acids, and that's saying, okay, well, we made enough. We have enough fatty acids, so stop making fatty acids. Use that acetyl-CoA for something else. Put it back in the Krebs cycle. Maybe by now, remember, the acetyl-CoA came out of the Krebs cycle because it itself inhibited pyruvate dehydrogenase and stopped glycolysis from happening, or stopped glycolysis from progressing to the Krebs cycle. So now it's here finding itself in fatty acid synthesis. Now it's done some fatty acid synthesis. Palmitate is saying, okay, that's enough. Maybe now we need more energy again. Maybe it's time to run the Krebs cycle again. So we just channeled from switched from one shunt to another, from fatty acid synthesis back into energy production. Okay. So this is just showing the same thing, just kind of more close up here, right? This is kind of going like yada, 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 past glycolysis and pyruvate and getting into our acetyl-CoA. So, just going right from glucose right to acetyl-CoA. There's our acetyl-CoA carboxylase, creating malonyl-CoA out of acetyl-CoA. And from there, we're going to do fatty acid synthase, or synthesase, and then elongation, cycles happening. And we end up with, say, any kind of fatty acid, depending on how many elongation cycles we do, but maybe we just start with palmitate, and that will go back and inhibit this step, and then acetyl-CoA maybe goes back to the Krebs cycle to create energy at this point. Okay, so we just switch from one to the other, and then as acetyl-CoA builds up again, as we start making more and more energy, then the acetyl-CoA will itself inhibit pyruvate dehydrogenase and stop the Krebs cycle from running, stop the production of pyruvic acid, and excess acetyl-CoA will come back, and we've maybe run low on palmitate by then, and we do this process again. So just back and forth, just switching from one thing to the other. Now this picture is showing how we go, how more of those processes are linked up. This is a neater version of what I drew earlier at the beginning of this. So here, normally we have glycolysis coming down over here, but they have glycolysis kind of going diagonally over here. So just always start at glucose. Here we have glucose going into glycolysis, pyruvate, into the Krebs cycle. There's the Krebs cycle being kind of a square there. They show the shunt of pyruvate fermenting into lactic acid. That would be heading then off to the Cori cycle, to the liver, right? To be reconverted back into pyruvate when we have more oxygen or more ability to run the Krebs cycle. Here we have the shunt of the pentose phosphate pathway. Remember, that creates NADPH, which we use for fatty acid synthesis. Here we have our acetyl-CoA and our citrate going out of the Krebs cycle, going into fatty acid synthesis. Remember, acetyl-CoA carboxylase is one of those enzymes there that will be pushing it along into the fatty acid synthase and the elongation and palmitate, etc. So all our different fatty acids, which will then go back and inhibit itself. Here we have oxaloacetate coming off of that initial process to go back into malate. So we see oxaloacetate and malate along with alpha-ketoglutarate and glutamate a million times here, how tied up with everything they are. But going back here into pyruvate, which can go back into, the Krebs cycle or up into gluconeogenesis. What it's not showing is here that we can do glycogen production, so glycogenesis. If we were to just take our glucose and go the opposite way here and do glycogenesis, we could do that too for more stored energy. And also amino acid metabolism isn't shown on this picture, so we could have amino acid metabolism coming in here. And again, just everything just so tied together and dependent on each other. And again, as if you didn't get the point that what we're talking about here is really just how everything is linked, and just to see the big picture here. Rather, we've been focusing on very small details for this entire semester. So I want to just, finish off with not as intense details and more looking at just the big picture and how everything is interrelated. So here it is, all the same stuff again, a little more different wording, same idea. There's our Krebs cycle. Always just look for that circle, look for the glucose and the Krebs cycle, and then branch off from there. We see our old friend, the urea cycle, with our glutamate, our glutamine going into ornithine and urea. Here are other amino acids branching off from that. Okay. So we've seen the ornithine cycle for urea, but we didn't look at this one this semester. All this different stuff we could do off of that or related to the ornithine cycle. The ornithine cycle is just one little part. We have a whole lot more we can do over here. Here we have fatty acid synthesis and beta oxidation going the other direction. Over here, even more amino acids coming off of just plain old pyruvate. From there we can go into even more proteins and amino acids, all kinds of different carbohydrates, not just glucose, but other sorts of carbohydrates. All of these with their own enzymes, all interwoven together. Okay, so that is metabolic channeling, just the idea that everything is tied together and is in a direct response to the other things. So any pathway that you have is going to affect all the other pathways that you have. So they're all dependent on each other. And this is going to lead us into what happens to our metabolic pathways in a liver that is fasting. So when you haven't eaten for a while, right? So you are running low on energy, need to break down things like fats and proteins to create acetyl-CoA and ketones, keto bodies. Remember, the brain can only use some very specific things for energy. This is very important. The brain only uses carbs for energy, ketones or keto bodies, keto acids, and also lactic acid for energy. So if you are low on energy, essentially low blood sugar, low carbs, you gotta get, your brain isn't having these carbs here. Lactic acid, that comes from pyruvate, so you're low on energy. You can use some lactic acid, but you have to have pyruvate, which, technically came from carbohydrates in the first place. This isn't a long-term solution. Ketones will be a long-term solution for the brain. So when I say long-term, I mean you're in the starvation phase for a certain amount of time, not just a typical, I've been asleep for 8 hours and I still have some lactic acid that the brain can use. You need to have these ketones if you start to go for longer than that without eating. Okay, so what you got to do is you got to break down your lipids and your proteins. You got to do gluconeogenesis to create glucose. So you have your ketones being created as you break down your proteins. You have beta oxidation occurring to run the Krebs cycle and create energy in other cells, but you got to get those blood sugar levels up in the rest of the body, or in the to serve the rest of the body and the brain. So gluconeogenesis and ketogenesis are occurring. Now remember, as you do ketogenesis, as you're breaking down your proteins, you end up with urea, so you end up using the ornithine cycle. So that's tied in there too. Now the ornithine cycle takes ATP, we saw that in the previous video, and that ATP is coming through beta oxidation. So here's where beta oxidation, ketones, ornithine cycle, all of that links together, and then we run the Krebs cycle to create energy from the beta oxidation, or the acetyl-CoA that came from beta oxidation. So all tied together. Okay, so because they are all tied together, anything that is causing a problem with one, or where one isn't working, is going to affect everything. So they all need to be working together on the same page, complementing each other. If any one of them is messed up, then it's going to start a cascade of effects. Now, a lot of your cells in your body are able to withstand that kind of thing. They're not as delicate. They may have lower energetic needs. They may be able to use, cells that can do beta oxidation or ketogenesis on their own and have, lots of stored energy will be more resistant to such things. But cells in your brain, they're not particularly good at utilizing other sources of energy. They pretty much need glucose, lactic acid, or ketones. And the liver, and also they have high energetic needs. The liver also has high energetic needs. It can do beta oxidation, but it still needs to have that really steady source of energy. So they're specifically extra vulnerable to the kinds of damages that can happen when any of the metabolic pathways are thrown off, or if they're deprived of their energy sources. So one particular source, this is a rare source, but it is something to be aware of if you're planning to have children especially, or if you work with children, and it's something called Reye's syndrome, and it's something that children are particularly susceptible to. Technically people under 18, but mostly people under 12. As you get older than 12 years old, then it starts to be less common, even though it's quite rare anyway, but even more rare after you're 12, and after you're 18, even more rare. So what Reye's syndrome is when someone uses aspirin to treat a child who is recovering from a viral illness like a flu, chicken pox, possibly COVID. I'm not sure if they've even had any reactions from that. I'm not sure about that. But a serious illness combined with aspirin can occasionally, rarely, result in Reye's syndrome for a child. And the reason why is that as the body breaks down the aspirin, some of the byproducts from that inhibit a particular enzyme that we use for beta oxidation. When you can't do beta oxidation, you're messing up all your other pathways. Without beta oxidation, you're unable to create your ATP from your fatty acids. so you're not doing the Krebs cycle there. Without that ATP, you have to do the ornithine cycle, or you can't do the ornithine cycle. So you're building up urea and ammonia. So you can do the ketogenesis, that's not messed up. You still got your ketones, but you're just being left with all this ammonia here, and you have a lack of energy. One of your prime energy sources is being cut off with Reye's syndrome. So Reye's syndrome is essentially very much similar to those metabolic disorders that we saw that had to do with a deficiency in beta oxidation. Any kind of problem with beta oxidation would have the same problems. And with Reye's syndrome, remember the damages from that were pretty severe. It could end up in brain damage, it could end up in swelling of the brain, inability to move, all kinds of problems, toxicity from the liver, all kinds of things happening just from, not being able to do beta oxidation. So whether that would be from a metabolic syndrome or from Reye's syndrome, either way, same thing, not good news. Now, why it doesn't really happen in adults, I mean, it does happen occasionally in adults, very, very, very rarely. The whole thing is super rare, but I don't believe that they know why it happens in adults. I'm not sure that I've seen any research that has gone into why children would be more susceptible. Maybe one day someone will look into that more. But so, like we said about the symptoms of Reye's syndrome. So can't do gluconeogenesis. So you get hypoglycemic. That causes problems with your brain that has the ketones, but not the sugar that it needs. Your muscles would be getting weak. Also, they can use ketones and stuff, but their fast source of energy is removed. Not being able to do beta oxidation, that's what I really focused on. So this can cause an inability to process your ammonia, like we said, with the inability to do the ornithine cycle for urea. Also, just having those fatty acids just accumulating, ready to be beta oxidized, but not able to be beta oxidized, so you're just getting like a fatty liver. Too much fat in the liver is going to lead to inflammation and possible liver failure, and once liver failure happens, that's a pretty bad situation. Now, even though you can do ketogenesis, as things start to back up, it'll slow down your production of keto bodies or ketones as this progresses. So as you start to create liver damage, your ability to create ketones is going to be lessened. So that would be kind of down the line a little bit. And when you can't make your ketones and you're still in starvation mode, then, well, now your brain doesn't have anything to use as energy except for lactic acid. So you're really causing damage to the brain. Okay. Okay, so those are the symptoms of Reye's syndrome. And like I said, any of those other disorders that we mentioned before that could impair your beta oxidation is going to have the same kind of symptoms. So, as you go from a fatty liver, the liver becomes fibrotic, and then it ends up in cirrhosis. There's lots of different causes of cirrhosis. Could be metabolic pathways. These are all really, really rare. Those metabolic pathways that any kind of deficiency in the metabolic pathways that deal with beta oxidation specifically are all pretty rare. But any one of the, pathways can affect all the other pathways. Beta oxidation is one of the more dramatic ones, but if you have trouble, if you can't metabolize glucose, say, we saw some situations there where you can't metabolize glucose, well, then you're stuck only doing beta oxidation and ketogenesis. So that's not an ideal situation either. So you need all of your pathways to function and to interact properly. So it's really pretty intricate. And there's so much more to cellular metabolism than what we've talked about this semester. So many different linkages and so many different enzymes and pathways that are all tied together. You could go into this forever, essentially. It's tied up with, pharmacology, how drugs work. It's tied up with, medical science, how to treat diseases, and how one disease can lead to another and can lead, to other, links between diseases. It's tied to nutrition, how much, antioxidants you're ingesting, or the fuel that you're giving your body, how your body reacts to each different thing, or toxicity, any kind of toxins that you ingest, how all that sort of stuff is tied together. There's so many different directions you can take with cellular metabolism, so many different things you can think about with cellular metabolism and connections. So we've just really barely scratched the surface with it. So hopefully you found it interesting, and maybe you want to look into it more and become a researcher about it, or go into the medical field and apply, learn more about specific areas and apply what you've learned to actual, medicine and stuff. But that does it for our videos. And that's it. That's our last video for the semester. Bye, everybody.